Thank you very much, George, to kind of you. I would like to thank you, the organizer, George and Alessandra, for the invitation and the possibility to share our work about self-renewal, clonal architecture, and tumor heterogeneity in pancreatic cancer, and what we learned in the last few years using molecular barcoding. So let me just outline a little bit the talk. So I'm going to talk about the generation of clonal tumors, a platform that we developed and published a couple of years ago, and the application of CRT to study treatment response in pancreatic cancer. Then I will talk about the generation of longitudinal CRT models to study unperturbed tumor expansion in vivo and also metastasis. This is an unpublished story. And then I'm going to show you some, let's say, preliminary data about the use of longitudinal CRT to study tumor microenvironment and the response of tumors to immunocheckpoint blockade. So first of all, let me acknowledge the people that actually were key for this project. So specifically, we started to use barcoding a long time ago with Alessandro Carugo and Seth Sahil when traction was not even traction, was CCT probably was not even CCT, so long time ago. But then, I mean, I was really lucky to work with three exceptional students in my lab and Giulio's lab. Alex now is not in my group anymore. Eileen, Nick, and also we were really lucky to work with Delia. She is a young biostatistician in the biostatistic department. She take care of a lot of mathematical analysis and modeling that actually were key for the generating the data I'm going to show you. So, as George said, my lab is focused on pancreatic cancer, and we are using mainly models that are derived from low passages PDXs and also mouse models like KPC models. So all the data I'm going to show you are generated mainly in vivo on these models, human or mouse. PDAC tumors, although they have a very relative simple genomic landscape that actually is characterized by dominant mutation of KRAS that are present in virtually all the patients. A very large fraction of patients is also mutation of p53. These tumors are highly heterogeneous, and the models we are using, they are able to capture this heterogeneity. So for instance, here I would like just to show you some single cell analysis, copy number variation or RNA-seq made on primary human cells that we are using and that I use also later on for some experiment. You can clearly see that actually at the genomic level, they are characterized by different subclones that can be also captured at the RNA level as different state of transcription. So I have really not to spend too many words to introduce tumor heterogeneity to this audience. You know that tumor are complex tissue characterized by genomic and functional heterogeneity, and that they constantly adapt and evolve in response to environmental perturbation that could be intrinsic, so changes really in the environment over time, and also extrinsic, like therapeutics. So we have an excellent tool to study heterogeneity. We have multi-regional sequencing, we have longitudinal sequencing, we have single-cell sequencing, but still we have a little of this understanding of the entire functional complexity of tumors in vivo. And this is the reason why we started a long time ago to use molecular barcoding. What is it, molecular barcoding? Molecular barcoding is a tool that enables us to study complex population dynamics in vitro or in vivo, okay? So our short stretches of DNA that we can insert in tumor cells or other cells, and these barcodes are inherited by the progeny of the cells when the cells divide, and obviously through sequencing we can study the composition of the population and getting data about the relative abundance or absolute abundance of different population of cells. But we get more because through the analysis we can really so the data can really infer about the replicative potential and the hierarchical organization of the original population of cells that we barcoded. I wanted to try to explain you how we generated clonal replica tumor, but I realized that actually it took too much time. I removed 10 slides just for the sake of time. But so we find a way to basically use barcoding and barcoded cells to generate large cohort of animals that bear human or mouse tumors with identical clonal composition, something that we named clonal replica tumors, okay? So here we have an example that we published some years ago. We have 3 tumors in a cohort. Each line here in the heat map is representing a barcoded population. And you can clearly see that actually the number of cells and reads generated by each barcoded population is exactly the same in the 3 animals, okay? Indeed, the correlation, the barcoded population is very, very high, is close to 1, so are virtually identical. And the common clones, the common barcoded population, basically are representing virtually the entire tumor mass. So we demonstrated that actually this is a very robust platform, is reproducible, and we can track the same lineages across different animals. This is really a key advancement of the platform, and we can do this almost with a single cell sensitivity. And so we can really run studies that we think are clinically relevant, and we can also do functional characterization or individual lineages that actually can be isolated. We at the beginning used the clonal replica tumors to run a preclinical study at the clonal level. So we randomize animals for different treatment using drugs with a completely different mechanism of action, like gemcitabine chemotherapy, a PI3 kinase inhibitor, or a MEK inhibitor. And you can clearly see that actually more or less you have a similar volumetric response using different drugs. But when we look at the clonality of the tumor, we found that actually the tumor clonal architecture in response to the treatment is quite different. So for instance, here we are displaying each clone that actually is found in the CRT. So each dot is representing a barcoded population. And you can clearly see that using gemcitabine, for instance, all these clones are basically depleted by the treatment. In red, you can see the clones that actually are gaining fitness and are selected by the treatment. You can clearly see that actually these are the clones that are spared by gemcitabine, get fitness, and basically sustain a tumor relapse. And these population of cells are completely different from the clones that actually are surviving the PI3 kinase treatment or the MEK inhibitor. What we learn is that we can really use drugs to perturb the system, perturb the tumors, and uncover heterogeneity. So for instance, just using 3 drugs, we were able to identify clonal lineages that actually were resistant to all the possible combination, okay? And this can really easily explain why these tumors have a very poor prognosis. One of the major advancements of CRT is that we can isolate clones with different response in vivo to gemcitabine, for instance, that are treatment naive. Okay, so basically starting from the cells that we use to generate the system, the barcoded cells. We can isolate virtually all clone of interest and then perform characterization. For instance, here we isolated clones that actually were spanning in vivo the gemcitabine sensitivity, the entire spectrum of gemcitabine sensitivity. And again, so obviously they have a different feature and they respond differentially to gemcitabine. So we performed deep characterization of these clones. We were not able to identify any genomic aberration that actually was explaining their differential response to treatment. We found very interesting things when actually we study the clones at the transcriptomic level, because we uncover new vulnerabilities. So one of the advantages of using this approach is that we can prioritize lineages isolated based on their behavior in vivo. Okay, so we know where actually are the resistant clones or the sensitive clones, and we can use these to prioritize basically the pathway analysis that we generate. Okay, and so we were able to identify that the baseline in cells that actually were never exposed to any chemo agent, basically the pathway that actually were upregulated in those clones and characterized those clones. And many of these pathways were druggable, and we demonstrated that actually we can eradicate these clones, as I show you in a few minutes. Another interesting thing, just to highlight the relevance of the approach, is that just basically getting the top 100 genes that were characterizing resistant clones and the top 100 genes that were characterizing the sensitive clones, we were able to generate a signature that were not curated at all, but we're able to basically dissect the patient PDACA cohort in TCGA and were predicted for their survival. So we were really able to recognize patients that actually had a very poor prognosis, probably just because this signature were able to detect pre-existing resistant population in these pre-treated tumors, because this patient in the TCGA never received treatment at the time of sequencing. Again, so part of this signature is made by pathway that actually are can be targeted. And so, for instance, we validated some, we validated DDR, DNA damage repair, but we also validated all the pathway like oxidative phosphorylation, that actually is one of the major features of the cells that are resistant to chemotherapy. And so we use again CRT to demonstrate the effect of the combination of OXPHOS inhibitor and chemotherapy on tumor clonal complexity. And specifically, we use here the IX-10759 that is a strong potent complex 1 inhibitor developed in the institute. So we use CRT, we again randomize it for different treatments, gemcitabine, IX, the combination. And you can clearly see here that again, we have the clones that are resistant to gemcitabine and sustain tumor relapse. And these clones are really targeted and disappear when we use the combination of an OXPHOS inhibitor. And again, so we were able really to prove that actually this strategy is able to eradicate clones and increase the survival of the animals. All these studies that represent the first generation of CRT have been done basically comparing untreated and treated tumors after the relapse, okay? And so they represent just a snapshot of what actually is happening in the in vivo. So we try then to generate longitudinal CRT that can really allow us to explore the dynamics of different population of cells during tumor expansion. And we use also to study metastasis. So Alex in the group basically generated a cohort of CRT and then started to sample animals at different time points over 3 months. And he found that this tumor evolved over time, maintaining their correlation until late time points when actually we have a drop. But still what is interesting is that for each time point, when actually he was comparing the clones that were in common between tumors, he found that actually these clones were representing virtually the entire tumor mass. And so we spent a lot of time working with Delia specifically and with our in Kim Do department to run a simulation of similarity and progression based on very stringent criteria that and doing a lot of complicated mathematics that really I barely understand. But at the end, we were able to validate with a very robust statistical approach and modeling the CRT, okay, and demonstrated that actually they progressed similarly until late time points. Specifically, what we found is that we can use these animals really as interchangeable variables, so we can really reconstruct the evolution of the tumor, capturing also some degree of natural divergence in the tumor over time. For instance, I would like just to show you here that a plot in which we basically are displaying the differences in distance, in percentage of barcodes between 2 animals in the group over time. You can clearly see that actually at very late time points, the vast majority of the clones are absolutely identical in terms of behavior. They are generating the same number of cells in 2 independent animals in the cohort. We have some divergence. For instance, here we have 6 clones that actually are growing, specifically expanding more in one of the 2 animals. But again, so the difference in terms of number of cells generated is minimal, is below 5%. So this is a very potent platform, and Alex decided to use to study metastasis and metastatic cascade. So as you probably know, the metastatic cascade is a model that has been developed a long time ago. And again, interestingly, the quantitative studies done were done using very artificial models that actually were selected repeatedly selected for the potential to metastatize. The model still is valid, absolutely, but we wanted to re-explore, and we were lucky to have to find a couple of PDXs that derived from a primary tumor in the pancreas. They basically recapitulated the disease in the patient, and specifically they maintain the same organotropism of metastasis. We had a model that actually in the patient developed specifically liver metastasis, and again, this is maintained by the PDX. And another model in which the patient succumbed and developed lung metastasis that also is recapitulated by this model. So we barcoded both, we use both, but I'm going to show you specifically data using the liver model. So what Alex did was to generate a cohort of CRT following over time, and over time sampling the pancreas, the blood, the liver, the lung of different animals, and then trying really to reconstruct the entire evolution of the barcoded lineages and the tumor at the body level. And so this approach is absolutely quantitative because we use a scale that allow us to convert reads in number of cells. So this is a picture of a quantitative picture of what is happening in these animals during 3 months. So the number of cells here, so the scale is not linear. Sorry about that. Here we have something like 300 millions. Here we have something like 300 cells. So, I mean, just be careful when you look at the graph here. But interestingly, what we see is that the tumor is growing, the orthotopic tumor is growing. At some point it starts to collapse. We have a lot of necrosis there. We are losing tumor mass. Over time, we see an increase of cells in the blood. But I mean, again, so we were, these are really few cells in the order of tens. Just we have a very late time points, few hundreds of cells. We have colonization of the lungs. As I said, this is mainly a model that actually developed liver metastasis. So the number of cells in the lung was not really high, it was maximum 1 million, something like that. We had a massive localization in the liver. And we also studied the ascites. That is really interesting because, I mean, in terms of burden, is really similar to what actually we found in the primary, okay, with hundreds of millions of cells. Interestingly, when we look at the clonality and the clonal complexity of what actually we found in different organs and tissue, we found that in the primary tumor, the complexity was maintained more or less over time. We can appreciate that in the blood, we had a very few clones that actually were detected over time. And we have a little bit more clones in the lung and in the liver, but again, so less than 1% of the total lineages that was present in the primary tumor was detected in the periphery. Again, an exception is the ascites, in which the number of clones is much, much higher. And this is recapitulating a little bit what actually was found in the primary, okay, with the very high complexity. So we perform clonal dynamics studies in different organs, and here I'm representing what is happening in the primary, and we found an unexpected degree of complex dynamics that naturally occurred during tumor expansion over 3 months. Okay, so each cluster here is representing behavior of specific clones in the tumor. And you can clearly see that actually this is a very complex behavior. So we have clones that actually are getting fitness over time and then lose fitness. We have clones that lost fitness and then reacquire fitness and so on. Something that actually Alex named alternating clonal dominance. And this is a behavior that we saw in many different tumors, in different models, and also in the mouse. So meaning that this is something that probably is happening in reality. And we think that actually this is due to changes in the microenvironment, okay? Also, although the expansion of this tumor is unperturbed because we are not using any external pressure, we know that actually these tumors are growing in a constrained environment, okay? And so many things are happening over time. But this is interesting because it's telling us that over time, when we inject cells in the animals, there is a continuous reshuffling of different population and obviously the clonality of the tumor. And again, so running probably experiment at week 4 will end up with completely different results than what we will get if we run an experiment at week 10 or week 12. Another interesting observation when we look specifically at very low representing clones in the tumor that were able to persist throughout the entire study is that many of these clones are really constantly attempt to take over the system, but very few success in colonizing the tumor, okay? So then we look at the clonal dynamics in secondary sites, specifically liver, and we found things very interesting there. So here we were just performing simple analysis, basically comparing the abundance of clones at week 12 with the abundance of clones at week 4, okay? And we found that actually we see many clones that basically sustain just transient seeding or colonization of the liver. So they are present at the beginning, okay, but then over time they disappear. Interestingly, it looks like that actually the vast majority of the clones that actually are able to seed in the liver never ever grow, and so they are there from early time points. But I mean, at least in the window that actually we observe, 12 weeks to 3 months is enough for animals. These clones basically are not changing at all. So we think actually these cells are exhausted, and we will talk a little bit about this later. Interestingly, we have another pattern of growth that actually is what actually really sustaining colonization and large nodes in the liver. So these are clones that basically sit in the liver very early on, and then they remain silent for several weeks until the moment that actually they really explode. And they form large nodes. The interesting thing is that the vast majority of these clones appear to be synchronized, and we do not really know what actually is happening. But they have a pattern of growth that actually is really reminding us the dormancy, okay? This is important because these are a minority of clones. So the vast majority of the clones that actually we found in the periphery, basically they do not grow at all, they do not expand. And so when we study seeding, metastasis, and dormancy, we have to be very careful because the vast majority of these cells probably really are not able to undergo any kind of reactivation. So at the end, it looks like that the metastasis outgrow pattern is simple. So we have a stable pattern at the beginning, then is followed by the explosive expansion near the late time points. And again, so this clonal dynamics at the secondary site here in red is not mirrored necessarily by the dynamics in the primary in blue. So for instance, for transient metastasis, you can clearly see that actually we have clones that actually are collapsing in the liver but are expanding in the primary, or vice versa, clones that actually are sustaining the outgrowth in the liver basically are disappearing or have a loss of fitness in the primary tumor. We try then to understand what was the driver of metastasis, right? What actually was really influencing the behavior of these cells. And we found that clonal abundance, so fitness in the primary, was correlated with the metastatic behavior. So you can clearly hear the metastatic, the clones that actually are able to sustain metastasis, that they are, in average, more represented than, more abundant than the clones that actually are not able to metastasize. But there are many exceptions. You can clearly see that actually here we have clones that actually are very, very low represented throughout the entire study that are able to sustain metastasis, or vice versa. We have clones that actually are very well represented and they will never be detected in the periphery. So we try really to understand a little bit if there were intrinsic property that were responsible for metastasis formation and dissemination rather than just clonal abundance. And to address this question, Alex and Elaine did a very complex experiment that I try to explain here, But they try really to design an experiment that was able to get rid of, the abundance, okay? The effect of the abundance. So what they did, they got patient-derived cells where they barcoded, and they passaged for a very minimal number of passages in a way that actually more or less the number of cells, of barcoded cells for each barcode was still similar, okay? There were no major differences between the barcodes. And then they split the cells and they transplanted orthotopically to study metastases. And the other half of the cells were injected in animals, and they perform serial transplantation over 6 months, okay? This is an assay that measures specifically self-renewal, okay, is able to identify the cells that actually have self-renewal and can have a long-term proliferating capabilities and can sustain the tumors in vivo for a very long period of time. And then they cross the two data sets and they compare the lineage. And what they found is that over passages, so they started from a very high complex population of hundreds of thousands of barcodes. Over passages in vivo, there was a decrease in number of cells that was able to sustain the tumor growth. And then, they compared the F4, so the tumors after the first cycle of transplantation with the clones that basically were sustaining the metastases in the liver. And they identify 187 barcodes there. So a very small fraction of the entire, the population that was injected at the beginning, okay? And obviously by definition, these 187 clones, population found in the F4 and contributing to the F4 have self-renewal. So interestingly, these red clones were responsible for virtually the entire metastatic burden in the liver. So 98%, more than 98% of the cells in the liver share these barcodes. And also they were responsible for a very large fraction of the F4 tumors, up to 94%. So meaning that, and these cells, as I said, were representing less than 0.1% of the original population. This experiment really is demonstrating that lineage intrinsic self-renewal property is an important factor for metastasis. Then we isolated clones again, as we did before for different treatment. Here we isolated clones that actually have a different metastatic potential. And we, again, did a characterization. We were not able to identify any genomic events that was justifying the metastatic potential. We then analyzed the transcriptomic level. We found a bunch of pathways that actually were specifically upregulated in metastatic clones, but specifically we focus our interest on pathways that were strongly deregulated in metastatic clones that were related to neuron and synaptic signaling, okay? And specifically, we focus on ionotropic glutamate receptor that specifically seems to be upregulated in the metastatic clones. So we validated, first of all, the phenotype of the clones. You can clearly see that actually the metastatic clones invade more in vitro, and when transplanted in vivo, they were able to sustain tumor growth, and specifically the metastatic clones were those that actually were able to colonize the liver. We validated the expression of glutamate receptor on these clones, and we demonstrated that the calcium intracellular level in the metastatic clones was higher at the baseline with respect to non-metastatic. So meaning that actually probably these receptors were functional, and indeed these cells respond to the glutamate. They were glutamate, exposure to glutamate increase their invasion in vitro, and they use an inhibitor, memantine, that actually is used in the clinic to treat symptoms related to Alzheimer, was able to decrease the infiltration of the cells, the invasion of these cells. So we used in vivo using clones, Pancreatic clones, and we were able to demonstrate that after 8 weeks of treatment, so we injected the animals orthotopically, and 3 weeks later we started the treatment, we were able to demonstrate that memantine has a strong effect in suppressing the burden of metastatic cells in secondary organ. So just to summarize this part, that the manuscript is in preparation, all these data are unpublished. So we demonstrate that, again, so metastatic cells can disseminate early after injection in vivo, and we demonstrated that exists population of cells able to see the distant organs, that at some point they lose fitness over time and disappear. This was a little bit unexpected, because it looks like that we have waves of seeding in distant organs. And then we use clonal dynamics. So we study clonal dynamics in liver and primary tumors, and we found that in the liver, the clonal dynamics are much simpler than what we found in the primary tumors. We have just a latency period that is followed really by an explosive expansion of clones versus really complex behavior like alternating dominance. And we demonstrated abundance and fitness is not the only factor that correlate with metastasis, and probably intrinsic factor like a long-term self-renewal can be critical for metastatization. The other thing is that we demonstrated that glutamate ionotropic signaling can drive invasion and metastasis. We have, I can see here, I had another slide that actually I wanted to show you. Let me see if I can get it, but I don't have, sorry about that. So let's move forward with the part 3. Then we try to use the longitudinal CRT to study the tumor microenvironment and the response to immunocheckpoint blockade. So for this experiment, obviously, since we need to work in immune competent microenvironment, we use mouse models. So we generated CRT from KPC barcoded cells. And obviously, we injected these cells in immunocompetent animals, Black6 immunocompetent animals. We did an experiment that was a little bit more complicated. I show here just part of the data for the sake of time. But again, so we had 2 groups. We had the treated and untreated. We sampled the treated over time to really then try to analyze the dynamics of clones. We found that, I mean, as expected, the anti-PD-1 treatment had some effect in terms of tumor volume with the reduction that was at least, was not minimal, but again, was not really strong. And again, so we confirmed that the treatment was able to increase the presence of CD8 positive cells in the tumor. Interestingly, what we found is that despite really the effect on tumor volume, the anti-PD-1 treatment had a dramatic effect on tumor clonality. And this was a little bit unexpected because we found that more or less 80% of the clones were depleted. by the treatment. We had more or less another 20% of clones that actually seems to be resistant and be able to expand during the treatment, and other clones, fewer that we consider no responders because they had minimal changes. When we look better at what was going on, we found that the shrinkage in volume was due to two main effects. One was due to the fact that actually we were really able to shrink the population of the lineages that actually were sensitive, okay? So we were losing, you can see here, probably something like 150 million cells. The other thing that actually we saw is that some of the barcodes that actually are sensitive really disappear and get eradicated. Interestingly, it looks like that the anti-PD-1 treatment, so the activation of the immune system, is really targeted cells and clones that actually are very highly represented in the tumors. Indeed, we evaluated that at least 60% of the tumor shrinkage is contributed by the most abundant clones in the tumor. And this again was something a little bit surprising to me. But we got very interesting things when naturally we look at the clonal dynamics of different population of cells with distinct response to the treatment. So again, here we are looking at treatment-naive tumors, okay? So we are basically using, we identify the clones that actually have different response after the treatment, but then, I mean, obviously we are just looking back in the untreated tumors, how these clones behave. And you can clearly see that the blue, they have, the majority of the blue, they have an upward trend, okay? So meaning that actually over time, over the different time points, these clones are really growing and they are colonizing the tumors, okay? They are those that actually are getting more expanded in vivo. On the contrary, the resistant clones, they have a downward trend. So meaning that actually they are outcompeted by the sensitive during tumor growth, and they have less fitness. So consider that actually both the clones, so the sensitive and the resistance, they are really unmasked to the immune system, right? Because they are able to survive. But again, it looks like that the PD-1 mediated immune system evasion confers some evolutionary advantage to these tumor cells, at least in this model. So then Nick tried to you know, he added an extra layer of complexity to the data and tried really to resolve the spatial interaction between the tumor microenvironment and different barcodes, the distribution of different barcodes in the tumor to identify basically the repetitive association between the stroma, the microenvironment, and the tumor clones that have a differential sensitivity to the anti-PD-1 treatment. And so to do this, basically, he and Lynn set up a protocol of laser capture, microdissection, and sequencing that actually allow us to, you know, detect barcodes multiplexing up to 300 different regions in the tumor. So this, I was happy because this decreased a lot of the sequencing cost. And in the meantime, on a sequential slide of tissue, Nick performed high content imaging using up to 40 markers to distinguish different population of cells. So from the study, the laser capture and sequencing, and we were able to reconstitute the spatial localization of the different population of cells in the tumor. And again, so we are looking here, and this is the beauty of the system that allows us to really identify in the tumor that is untreated where specific clones with specific behavior are and which region are populated by these clones. Again, so was the same plot I showed before. We just changed the color for just the sake of representation. But you can clearly see that actually the PD-1, anti-PD-1 resistant clones and the non-responder, they are especially localized in the tumor. They populate specific domains, microenvironmental domains, okay? They are not diffuse everywhere. They are specifically located in a specific region of the tumor. And so Nick used multiplex staining, trying really to identify how this specific region, the feature, the microenvironmental feature of these specific regions, okay? And so we, using this approach, he was able to demonstrate that we have a very high degree of spatial heterogeneity in the CRT that you can see here. This is just an example of other markers on the same region. Here we have myeloid markers or markers that actually are staining the microenvironment, the stromal component. But what Nick did that was unique was really developing an image data processing pipeline very robust that allows him to resolve imaging data at a single cell level, and he can really analyze this data set as we usually analyze single cell transcriptomic data. This was possible because, I mean, when actually he performed segmentation, he was really able to annotate each cell for the expression of specific marker, then can be really used as transcriptomic data. And this is exactly what actually he did. For instance, here he was able to resolve cell phenotype using UMAP. These data are not transcriptomic data, these are imaging data. And then again, and so just for the sake of representation, we can merge different population. And obviously, since actually we have the spatial information, we can reconstruct the entire structure of the tumor and know exactly where this population of cells are. Then obviously he can integrate this information with the clonality that actually detected through laser capture and sequencing of the tumor. And this is exactly what actually he's doing. So he's able to map interaction between cancer cells with different behavior and the tumor microenvironment at the clonal level. So here, sorry, I just mask the different signature because this is something that we are actively working on. But you can clearly see here we are evaluating the association. You can clearly see that, and this is normalized on the sensitive clones. You can clearly see that the peak above 1 are just indicating that those specific region with specific feature are populated by non-responder or resistant clones. You can clearly see that actually we see a peak in hypoxic region, we see a peak in regions that actually are populated by Treg. We see very interesting things like a depletion of resistant clones in regions of the tumor that actually are actively proliferating. This is in line also with the data we demonstrated before that sensitive clones are, they expand better and they have more fitness. So this is the kind of work we are doing, and so we are exploring all the other association, all the other texts, because we think actually we have interesting things there. And so just a brief summary for this part is we uncovered that the activation of the immune system has a radical effect on tumor clonality despite of the lack of effect on tumor volume. This is something that actually we see repeatedly in our models and using CRTs. And so, I mean, again, so if in your model you do not see a volumetric effect of the treatment, whatever it is, I mean, there you could still expect to have a huge reshuffling in terms of clonality. And then again, the other interesting observation is that the PD-1 mediated immune evasion confer for sure growth advantages to tumor cells, at least again in the model we used. So we are really in desperate need of cancer immunologists as a collaborator, because we need really to try to understand a little bit better all the data that actually we are generating. So, I mean, if you are willing to collaborate, just shoot me an email. So, yes, I would like just to acknowledge the people that actually did the work. So again, Alex, Eileen, and Nick, and Delia in the biostatistic department. And that's it. So I think actually I have, if you survive, still time to get some questions. Thank you for your time.
Thank you very much, Andrea. Wonderful talk, very, very exciting. And in fact, let's start with a question. So the first question, it's hello, Dr. Viale, very impressive strategy to use barcoding to select cell clonality. The question is, please explain better how the barcoded cells are sorted. If using fax, do the barcodes have a fluorescent reporter that help with sorting?
No, so we are just infecting tumor cells with a library, viral library that basically is inserting these short stretches of DNA in the cells. So everything that actually we did was through sequencing, next generation DNA sequencing. Okay, so we get the tumors or the population we are interested in studying, and we extract DNA, and we sequence the DNA. We generate libraries for sequencing to detect the barcodes. I have to say that some of these barcodes that we use, specifically in the last project about the study of the microenvironment, we use expressible barcodes. So those barcodes are detected, can be detected also at the RNA level, single cell sequencing, and this is exactly what actually we are trying to leverage as well. We didn't do it yet, by the way.
Thank you very much. Another interesting question by Sanjay Singh. So I really appreciate your presentation. The question is, if you take a metastatic clone, barcoded, and then you re-barcode this one and re-grew the tumor, would you expect the heterogeneity landscape of these new tumors to have similar patterns as the parental metastatic tumors?
This is a very interesting question. We didn't do yet, but I can tell you probably yes. So what we know is that the lineages itself that actually we are tracking are quite heterogeneous in a way that they are. So when we do, for instance, or also single cell sequencing, there are different clusters there. So you have a different cells with different behavior within the same lineage, because the lineage is really recapitulating a little bit what is happening when actually you have stem cells that differentiate, right? So you call with the same label, with the same barcode, cells in the same lineage that obviously have different properties, but they are clonal, so they derive from the same ancestor, okay? So yes, I expect heterogeneity, but still, I mean, specifically in those barcoded cells, we find the ability to metastatize, okay? That, again, is a minority of cells, so it's really a fraction of cells that is able to seed in the periphery. And this is a little bit counterintuitive, because you expect to have tumors that actually are shedding, and the shedding of the cells should be really not selective, okay? So you expect to find in periphery more barcodes that actually we really detect. And we do not know why, but it looks like that actually what is in the periphery is a very small fraction of the primary tumor, although the vast majority of the cells apparently are exhausted.
Thank you very much. Another interesting question by Dr. Walter Hittelmann. Hi, Walter, great having you here with us. So, did you see any evidence of clonal cooperative interactions between different clones?
Yes. And I can tell you specifically, this is something that we have to discuss with immunologists, by the way. In the last part, is something that we see. Many of those resistant cells are basically usually populating specific regions, but some are also present in regions where actually we have sensitive cells. So we think that actually, they probably share some kind of crosstalk. And this is the reason why the experiment we did is much more complicated, in which in reality, we do not just have two groups of treatment, but we started the anti-PD-1 at different time points, giving to the cells the possibility to reorganize their microenvironment. But absolutely, yes. So we see some sort of cooperation, but it is really difficult to. So we would like to work on this as well. But again, so we have limited resources. And this, I'm telling you, it's very difficult to get funded using barcoding. Okay. So I mean, that is a major problem, because these experiments are absolutely expensive. And so far, actually, I was able just to get one grant, a small one, by the way. Not another one.
So we wishing you good luck for more grants, because the science is exciting, and as usual, very exciting and advanced science. It's passing the capacity of reviewers of grants to get it fully.
At least now we are getting data that and phenotypes that obviously cannot be questions, and so maybe starting from there we will be able to secure some money. Let's see, and funds.
So with
congratulations,
this,
Andrea, very innovative. I just wanted to say congratulations.
Thank you, Alessandra. Too kind.
Yes, congratulations, Andrea. Thank you very much for your time, and thank you to all of you who attend. Again, to all of you, happy holidays, enjoy life, and let's make discoveries in 2022. And in fact, in January, we have another wonderful talk from Wenyi Wang. So we are looking forward, all of you, to January 6th, Thursday, for Enjoy Science. Thank you again, Andrea. Good luck with everything, and good luck to all of you.
Thank you very much.
Ciao,
Have a good day, everyone. Bye. Bye-bye.